Hi class, how are you? So this is technically the last lecture of the term. There'll be one more lecture for what would be Thursday's class, which will basically be a review where I go over everything we covered in the term. And so today we're just going to cover circadian rhythms and stem cells. So this technically won't be on your final because your book does such a horrible job, if any, of covering circadian rhythms. And although it does a decent job of the stem cells, it's all just sort of very peripheral. So I don't tend to put this on the final exam, but yet it's still really good knowledge, especially for those of you who are heading off to medical school and for you guys who are heading off to graduate school. So I think it's definitely worth your while to pay attention and understand this stuff. And just because none of the direct material will be questions on the questioned on the exam, there may be things that are alluded to it. But really, there's not much here for you guys. So just sit back and enjoy. Okay, so the circadian clock it's also called the biological clock, or it's often referred to as the circadian rhythm. And these are daily biological rhythms that control a lot of the gene expression in our body and really control activity and metabolism, the timing of development, and a whole bunch of other cellular processes. It's most obvious with the sleep-wake cycles in mammals. So, we are day active creatures, so we go to bed at night and then we wake up in the morning. And actually, what's interesting is your clock changes over time. And so when you're in your late teens and early 20s, your clock is such that you probably like to go to bed at 1 or 2 o'clock in the morning and sleep till 10, 11 o'clock in the morning. But then you'll all notice that your parents probably go to bed pretty early and wake up pretty early. And that's just because their natural clocks want them to get up early and go to bed early. So the word circadian actually comes from Latin. It's circa for being about, and dien being day, so circadian. So these are rhythms that are about a day. Now, in order to be a circadian rhythm, you have to have sort of 3 criteria have to exist. Okay, the first is that the circadian rhythm has to be entrainable. Okay, so typically the clock is reset by light or temperature. And for any of you guys who have traveled overseas and gotten jet lag, you'll know that that's basically just your biological clock resetting. The clock has to be self-sustaining. So what this means is the clock in its most basic form is an anticipatory system. It gives organisms a fitness advantage. So for example, if you are a nocturnal animal and you sleep during the day, if you want to be the first mouse out to get the scavengings of food, if you can anticipate the change in from day to night, you can get up right before nighttime, and then you can go out and get your food. So you end up with a fitness advantage. And so what that means is that self-sustaining is that the clock can run under constant conditions. So in the absence of any sort of external cues like light and temperature, your clock will continue to oscillate. So if you were technically thrown into solitary confinement, you would still roughly sleep and wake on the same cycle that you are when you were thrown in to solitary confinement. The final thing is temperature compensated, and this doesn't really apply much for homeotherms. So we're considered homeotherms because we maintain a core body temperature. However, there are a lot of organisms with circadian clocks that don't control their body temperatures, and their period changes regardless of the temperature. And why this really comes into play, it has to do with biochemistry. So typically rates of reaction are sort of proportional with temperature. So as temperature increases, the rates of reaction increase. Okay, so if the clock wasn't temperature compensated, if it ran faster at warmer temperatures than it did at lower temperatures, then it would actually cease to be a clock and it would be a thermometer. Okay, so you can sort of think of it like that. So regardless of the temperature, in sort of a sort of defined range, the clock runs at around 24 hours regardless of the temperature. So that's what temperature compensated means. It's compensated for any changes in the temperature. So one of the things about the biological clock is its implications in disease. And really the last 20 years of research has found basically almost every sort of disease has some sort of circadian connection. It's sort of started with some findings in cancer, which I'll talk a little bit about, but also metabolic syndrome and cardiovascular disease are two others that are, and also a lot of psychological disorders have a clock connection. But the disease connection all sort of started with the observation that tumor suppressors and oncogenes were oftentimes circadianly regulated. So P53 is circadianly regulated, MYC, and a whole bunch of other things that I covered last time. They're sort of all circadianly regulated. And this was sort of expanded when it was determined that the oncogene RAS as well as ROS, which is reactive oxygen species, they can feed into the clock to regulate circadian controlled gene expression, or CCG expression. The clock also regulates the timing of cell division, and there's feedback between the cell cycle and the circadian clock, and the clock actually gates the cell cycle. It says you have this window during the day with which a cell should divide. Okay, and that's what it means by the clock sort of gating the cell cycle. And the role of the clock in cancer is really an area of active research. And a lot of it comes from the fact that it is very physiologically relevant. Okay, so night shift workers, so nurses who work the night shift, actually have a much higher incidence of cancer compared to their day shift counterparts. And light at night is now considered a carcinogen by the World Health Organization. And in regards to cardiovascular disease too, airline pilots who are constantly changing time zones and getting jet lag, they have a much lower lifespan and a much higher incidence of cardiovascular disease than do the general population. So yeah, they have just a reduced lifespan. And you can recapitulate a lot of this stuff with mice in the laboratory. If you phase shift them on a regular basis, the mice will actually die off faster. So here's some key terms you need to know when you're talking about the clock. So the first is entrainment. That's what sets your clock. Okay, so typically light and temperature are the big entraining cues. And here you can see the sort of entrainment rhythm where you have light, dark, light, dark, light, and then you get to your free running rhythm where you can still see changes. And so this creates oscillations, and so oscillator is a big term. You also have a period, and your period can be chosen from any point. So it's from here, it's peak to peak, but you can also determine the period from trough to trough. It also has a phase, and the phase is relative to whatever entrainment cue that you're using. And then another thing is the pacemaker. We'll see that in a second. The pacemaker cells, which actually in mammals reside in this part of the hypothalamus called the suprachiasmatic nucleus, which is a mouthful to say, so from here on out I'm just going to call it the SCN. Okay, so the suprachiasmatic nucleus, it's right here in sort of the base of your brain. it's a group of cells in the hypothalamus, and what ends up happening is you perceive light through your eyes, and that transduces signals to the SCN, which help to reset the clock. And one of the things that the SCN does too, in conjunction with the hypothalamus, is it regulates rhythms in hormone release like melatonin and other things. But ultimately what happens is you end up with these sort of output rhythms, which are physiology, behavior, as well as, you know, just total changes in gene expression in the cell. You can measure circadian rhythms in a lot of different ways. Historically, one of the great systems was activity rhythms. And so if you put a running wheel inside a cage with mice and you sort of follow them over 24 hours, they will basically, they're nocturnal, so when the dark bar up here shows nighttime, they're active during the night, and then during the day when the lights are on, they're sort of sleeping. And here you can see that the first like 20 days or so here are on LD cycles, so light dark cycles, and then DD stands for constant darkness. And so here's they're being entrained, and here they free run. The data here is shown as a double plot, so here is day 1, here is day 2, and then over here is day 1. The next line down is day 2, day 3, and then day 3, day 4. And so every day is sort of shown twice except for the first day. Now, you can actually determine the period and the phase under free running conditions by sort of following the slope here or following the slope here. And as you can see, the ones down here, these actually sort of lack a clock. So this figure right here to the right was actually done in the 1990s when this group basically mutagenized mice to find ones that would lose rhythmicity. Okay, so they just did an EMS mutagenesis screen for a mutant mouse, and they called that mouse clock. Okay, and it turns out that clock was a mutation in one of the transcription factors, as we'll see in a second. All right, but all this data is actually real data from 1994 that came out of a manuscript. Okay, so the mechanism by which the clocks are controlled is a transcriptional, translational, positive and negative feedback loops. What I really want you guys to know is that the clock is really regulated by a transcriptional negative feedback loop. These other sort of loops are somewhat ancillary to the mechanism, but this core feedback loop here, the transcriptional negative feedback loop, is what's really important. Now, in a whole bunch of sort of genome-wide studies on the clock, there's estimating that roughly 40% of all the genes in a mammal are under clock control. Now, in any given cell, you might only have 10 to 15%, but if you look in all different tissue types throughout a whole organism, roughly 40% of all the genes are under the control of the clock. So for example, you could have a gene in your liver that's actually rhythmic in the liver, but you also have that same gene expressed in muscle cells, and it may not be rhythmic in muscle cells, but there could be genes in muscles that are expressed in the liver, and they're rhythmic in the muscle, but then they're not rhythmic in the liver. So what I'm trying to say here is the number is really high, but that doesn't mean that in every single cell 40% of all genes are under the control of the clock. Okay, in any given cell, maybe 10 to 20% of the genes, but if you look at all genes over all tissue types, rhythms exist in 40% of all the genes. Now, we've also seen through that last figure, the clock controls many physiological processes, activity rhythms, metabolism. There's a reason oftentimes when you wake up in the morning, the first thing you want to do is is go have breakfast. It regulates cell division, and there's also interconnected circuit between the clock and DNA damage. So if you have DNA damage, that arrests the cell cycle, and it also can feed back to the clock to sort of slow the clock down till the DNA damage is repaired. Okay, so you kind of need that if you want to think about if the clock is governing when the cell cycle can happen. If you have defects that are stopping the cell cycle, you also want to stop the clock so that once that gets fixed, the cell cycle can proceed and the clock is giving it the window with where it can proceed. So I hope that made sense. But here's basically the transcriptional negative feedback loop in its most simple form. What you have are positive elements. These are transcription factors that drive expression of clock genes and clock output or clock control genes. And then the clock genes themselves make up the negative elements that then go back and inhibit this. And we've already seen in a couple lectures where negative feedback can generate oscillations, and that's exactly what's happening here. And so in mammals, the positive elements are clock, which we saw from the mutant screen just a second ago, and this other transcription factor called BMAL1. Now the negative elements are the period genes and the cryptochrome genes, so PER and CRY. Okay, so clock and BMAL positive elements, PER and CRY are the negative elements. And ultimately these oscillations generate rhythms in metabolism and behavior and a whole bunch of other things. Now here's how the clock basically resets. You get an environmental input, and that can be sensed by the positive elements. They'll drive expression of the clock genes, which then form the negative elements, and then those negative elements sort of feed back to inhibit. At the tail end, there's also degradation of the negative elements, and once the negative elements have inhibited the positive elements, they actually get removed from the cell. And so in this, you have basically rhythms in all kinds of different species. Here's a bacteria, cyanobacteria has a clock. This filamentous fungus Neurospora crassa has a clock where there's rhythms. Flies have it, and insects, so Drosophila is a great model system. You have mice and humans, and also birds. So all of these different organisms, they all have a clock. And they're all basically set up the same way, where you have a negative feedback loop. Okay, so in the mammalian clock, the positive elements are clock and BMAL, and these are helix, loop, helix, pass domain containing transcription factors. So we've seen the helix, loop, helix protein and how the sort of the helixes fit in the major groove of DNA, and they bind to E-box elements. And then you have the negative elements, the period proteins 1, 2, and 3, and cryptochrome 1 and 2. The period proteins also contain pass domains, and CRY contains a flavin adenine dinucleotide, and clock is also, the transcription factor clock is redox sensitive. So it can sense changes in the redox potential. So here's basically how it works if you sort of look at it from a cellular level. So here you have a nucleus, and here you have clock and BMAL. They bind to promoters in the period 2 and cryptochrome genes, and then they will drive expression and transcription of PER and CRY. So then you get formation of the PER and CRY messenger RNAs. They've got a 5' cap and a poly A tail, of course. And then they get translated to form the PER and CRY proteins, and once you have the PER and CRY complex here, you get CK1 and CK2 forming. These are casein kinase 1 and 2, and they start to actually phosphorylate PER and CRY, and when they're sort of early in their phosphorylation state, they re-enter the nucleus and shut down their own expression. And then they leave the nucleus and become sort of hyperphosphorylated, and that releases a degradation signal on the period 2 protein, and it's degraded by the proteasome, okay, with the skip-cullen complex. It's a little more complicated than this, and the purr and cry complex is actually a huge dynamic complex that changes throughout the cycle, and when it's in the nucleus down here, it's actually recruiting chromatin remodeling enzymes and things that are going to shut down expression and basically make a non-permissive state for transcription. Here's just another schematic. These are often drawn as wire diagrams, and we've sort of seen these before, but you have clock and BMAL. They'll drive purr and cry, as well as reverb alpha and RORA, and these then go on and have a positive and negative effect on BMAL expression, so there's actually a rhythm in BMAL as well, and then the purr and cries come back and inhibit that. So there's additional loops here, but really what I want you to focus on is the primary loop, where you have clock and BMAL driving purr and cry, and then purr and cry inhibit clock and BMAL activity. But the reverb alpha loop over here regulating BMAL is also important, because it's really important in metabolism and lipogenesis and other things like that. Here's just some molecular rhythms that you can see. I grabbed these out of some old papers. Here's the rhythm in BMAL protein. There's also a rhythm in clock protein. There's a rhythm in cry. And these are basically sampled every 4 hours starting at 0. So 0, 4, 8, 12, 16, and 20. And you can see here that the PER2 and cry sort of peak in sort of the late night, and these ones sort of peak in the afternoon. If you take it out further, for example, what you have, and this is just a different experiment, where you have basically a rhythm in mRNA transcripts. Once again, these are sampled every 4 hours over 2 full days, and you get this sort of nice rhythm. And then you can also see changes in the clock proteins. This happens to be freak and neurospora, where it's started to be made. You can sort of follow it right after here, this 12, where the peak in mRNA is. All of a sudden, about 4 hours later, you start to see the peak in the protein, and it becomes progressively phosphorylated and then turned over. And then you can see the next round where, you know, the peak in transcription was at 32, protein builds up at 36, and then it gets progressively phosphorylated and turned over. So you have this constant thing where the rhythms in transcripts and the rhythms in the protein. So the clock and disease, specifically the clock and cancer. So the mammalian PER2 gene is considered a tumor suppressor. Mutations in PER2 have been detected in breast and colorectal cancers. There's also aberrant DNA methylation and histone acetylation of the PER2 gene in human tumors, and cancer patients oftentimes have sleep disorders. In regards to RAS in the clock, so it was discovered first in Neurospora that RAS, which is shown here in blue, and part of the RAS MAP kinase signaling cascade, this feeds directly into the clock. This red protein here is son of sevenless, so this is just the structure of it. And mutations in RAS, which we covered in the cancer lecture, are responsible for about 30 to 40, are present in about 30 to 40% of all tumors. And increasing reactive oxygen species or superoxide radicals also feed directly into the clock. And elevated reactive oxygen species are often found in tumors. And this has sort of a self-fulfilling problem, because the reactive oxygen species can actually go on and further damage DNA. And so there's an awful lot of crosstalk between RAS and ROS, and the health benefits of antioxidants are now clear. And actually, melatonin sort of serves as an antioxidant. So DNA damage in the cell cycle. So the clock gates the cell cycle, and WEE1, cyclins, and P21, they're all regulated by the clock. And the DNA damage response is also controlled by the clock, right? And so the DNA repair enzymes and all of those kinds of things as well. The checkpoint kinase, which we really didn't cover too much, is associated with clock proteins. But if you have DNA damage, the checkpoint kinases get phosphorylated, and they help to sort of shut down the cell cycle. And these can actually phosphorylate both freak and neurospora and period in mammals. And P53, like I said in the last slide, is circadianly regulated. And there's a substantial amount of research on this, and active research going on as we speak in this specific area. So there's this guy, his name's Stephen Hill, and he's down at Tulane Medical School in New Orleans. And he's got some really cool research, because one of the things that he's found is that melatonin, which is a circadianly regulated hormone and helps sleep. I'm sure you guys have all heard that, you know, if you're having trouble sleeping, you can take melatonin. It's sold over the counter, but it's a circadianly regulated hormone, and it has antioxidant properties. And oddly enough, it's been shown to arrest cancer cell growth in tissue culture and in animal models. And melatonin signals via the melatonin receptor. So this is just a really sort of an interesting thing, that it has such great anti-cancer properties. Unfortunately, the pharmaceutical and biotech industry has very little interest in researching this specific sort of thing, because there's no money in it for them. So it's just interesting to see how important academic research is in pushing ideas like this forward. There's also the thing with chemotherapy in the clock. So, and I mentioned chronotherapy before, and what people are finding is that time of day specific treatments can have a dramatic effect on patient survival. Okay, so chronotherapy. And the idea is that you want the chemotherapy to be sort of floating around when it's needed, and not floating around in your system 10 hours before it's needed. Okay, so the idea that treating a patient in the evening versus the early morning, that may actually have major implications on patient outcomes. So for example, let's say you want to have a chemotherapy that's treating a specific protein and targeting a specific protein or a specific cellular process that happens at 10 o'clock at night. If you're pulling that person into the hospital at 8 in the morning and you're giving them a whole bunch of toxins like chemotherapies, and they're sort of floating around in the body and being broken down by the liver, so that there's enough around still at 10 o'clock at night, you can sort of see if you just give it to them at 9 o'clock, it would have a much better effect, and you can actually use less drug, and that would cause less negative outcomes, right, and less side effects. So the idea sort of holds that there might be a best time of day to give a drug treatment. And I want everybody that's going to medical school to just keep that in the back of your head. Okay, when is the best time that you should be asking your patients, or telling your patients to take a specific medication? And with the knowledge that most drugs are broken down in the liver, and most of liver function is under circadian control. And they're finding this with all sorts of things from, cardiovascular treatments and a whole bunch of other things. So one of the things I didn't mention about the cardiovascular things is people tend to have heart attacks at a specific time of day. If any of you turn out to be ER doctors and you're on the morning shift, you're going to see a lot more heart attacks than you will if you're working the night shift that day, right? Most people have a heart attack when they wake up in the morning, and so most heart attacks actually occur between sort of 7 a.m. and noontime on any given day. So once you get past noon, you're usually in good shape. So that's it for now. We'll take a break here, and then in the next next part of the lecture, I'll go over stem cells.